Hello everyone. You are listening to this audiobook on channel Voice for Purpose by NSS IIT Bombay.
Amidst this pandemic, vaccines seem to be our only ray of hope and is a topic on everyone's mind. Vaccines give you immunity to a disease without you getting sick first. They are made using killed or weakened versions of the disease-causing germ or parts of the germ called antigens. When you get a vaccine, your immune system responds to the vaccine in the same way it would to the real germ. It creates antibodies in response that become part of your immune system. That way, if you are ever exposed to the disease-causing germ in the future, your immune system will be able to quickly destroy it before it has a chance to make you sick. This is how you get immunity from vaccines. The practice of immunization dates back hundreds of years. Buddhist monks drank snake venom to confer immunity to snakebite. In the 15th century, the Chinese recognized that people who had contracted smallpox once were immune to reinfection. They came up with the idea of preserving scabs from individuals who had suffered mild cases, drying them out, crushing them to a powder, and blowing them up the nostril. Edward Jenner is considered the founder of vaccinology. In 1796, he injected an 8-year-old boy with the cowpox virus. The boy then showed immunity to smallpox, which was a much deadlier disease, and thus the first smallpox vaccine was developed. Currently, researchers around the world are working on a vaccine against COVID-19. Presently, there are over 50 vaccines in different stages of development. Before a vaccine is given to the masses, it has to pass many tests and trials. The first step in evaluating a vaccine is animal testing. Animals are given differing doses of vaccine to check for adverse events and an immune response. Animal testing provides information about safety before the vaccine is tested on humans. Human testing in clinical trials is divided into 3 phases. In phase 1 trials, 10 to 100 healthy volunteers receive different doses of vaccine and their response to the vaccine is observed. Blood is drawn from the volunteers to assess the immune response. Phase 2 trials include hundreds of healthy volunteers who are more diverse to give a better indication of the safety and immune response in people likely to receive the vaccine in the future. Phase 3 vaccine trials are large randomized controlled trials conducted in the community. In these trials, people are divided into 2 groups. One group receives the vaccine and the other a placebo or a dummy vaccine. If fewer people who receive the vaccine become sick compared to those who receive the placebo, it is direct evidence that the vaccine prevents the disease. It is only the phase 3 trials that can provide a pivotal demonstration that a vaccine both works and is safe. There are 4 categories of vaccines in clinical trials: whole virus, protein subunit, viral vector, and nucleic acid. In the whole virus vaccine, either a weakened form of the virus or inactivated virus is used. Live, attenuated vaccine may risk causing disease in people with weak immune systems, whereas inactivated virus vaccines can be given to people with compromised immune systems. Subunit vaccines use pieces of the pathogen, often fragments of protein, to trigger an immune response. Doing so minimizes the risk of side effects, but it also means the immune response may be weaker. Nucleic acid vaccines use genetic material, either RNA or DNA, to provide cells with the instructions to make the antigen. Once this genetic material gets into human cells, it uses our cells' protein factories to make the antigen that will trigger an immune response. The advantages of such vaccines are that they are easy to make and cheap. Since the antigen is produced inside our own cells and in large quantities, the immune reaction should be strong. A downside, however, is that so far, no DNA or RNA vaccines have been licensed for human use, which may cause more hurdles with regulatory approval. Viral vector vaccines also work by giving cells genetic instructions to produce antigens, but they differ from nucleic acid vaccines in that they use a harmless virus different from the one the vaccine is targeting to deliver these instructions into the cell. One type of virus that has often been used as a vector is adenovirus, which causes the common cold. Viral vector vaccines can mimic natural viral infection and should therefore trigger a strong immune response. However, since there is a chance that many people have already been exposed to the viruses used as vectors, some may be immune to it, making the vaccine less effective. India's indigenous vaccine against COVID-19, Covaxin, is a whole virus inactivated vaccine. Phase 1 and 2 for Covaxin have already been completed. Encouraging Phase 1 and Phase 2 Covaxin trial results have paved the path for Phase 3 clinical trial in India, which is currently ongoing.
So hope you guys had fun. Please like, share and subscribe to this channel for more interesting audiobooks.